Hello and good evening, wherever you are in the world. Thank you for joining us for another one of these fascinating discussions. Today, it's going to be with Dr. Sabine Hazan about the microbiome. And believe me, when you hear who she is, you will be surprised that you don't know more about her. This is likely to be a really important discussion. Just a reminder before we start, as usual, click on the subscribe button. It's usually hidden, but it's now in the bottom left right hand corner of your screen. There'll be a little button there and you can subscribe. Additionally, I want to remind everybody about Humming Heroes. This is our book. We have 2 days to go before it's launched on Amazon. This is going to be fascinating. So use the link below to pre-order it, and we look forward to your support. So, getting straight into this important discussion about the microbiome and understanding a bit more about it is we've got Dr. Sabine Hazan. So, I'm going to bring her onto the screen so she can introduce herself. Sabine, how are you?
I'm doing great. Thank you.
Excellent, excellent. So, Sabine, tell us a little bit about yourself.
I'm a gastroenterologist. I've been practicing for almost 3 decades. And I started GI as a fellow in research. So I started clinical research. So I got into the world of pharmaceutical clinical trials for about 3 decades since fellowship. And from clinical trials, I realized, there were a lot of drugs that we were using for similar drugs for different diseases. So I got exposed to all that and started questioning, are all diseases similar in a way? Is it all starting in a gut? Is it all starting as our immunity? Especially with the advances with biologics that were used for Crohn's disease, for rheumatoid arthritis, psoriasis, etc. So what happens is from there, I entered the world of a clinical trial for C. diff. C. diff got us into the microbiome because it made us understand C. diff, a bacteria that causes people diarrhea and they can die. Made us, I started doing a lot of clinical trials on C. diff. And then from C. diff, the microbiome kind of exploded because of the fact that we started doing fecal transplant, the process of taking stools from a healthy donor and putting it in an unhealthy C. diff patients and curing C. diff. So from that, clinical trials became all about the microbiome and fecal material in capsules. And I questioned the whole field of, what are we doing here? What are we doing when we are improving Alzheimer's by changing the microbiome? And so I started sending my stools to different labs. And then from there, I realized there was no validation in the world of microbiome testing. And so I started my own genetic lab, really to understand, what does Alzheimer look like, et cetera, et cetera. And so here
Yeah.
we are, as the owner of a genetic lab, Progenabiome. And it's
Mm.
kind of funny that, because you're posting that, I put it as the quest of a new healthcare revolution. When COVID hit, I felt, I have a genetic sequencing lab that's understanding the microbiome. I have a CRO, a clinical research organization, that basically works with the FDA to bring, products to market. So who better than, and then I'm connected with Dr. Tom Barodi, who is the father of fecal transplant, and started a lot of patents on treatment. So I said, who better than me and Dr. Barodi to step into COVID and try to figure out COVID? And so really that's been my path. It's been an interesting road, and here we are.
Wonderful. Listen, now, one of the things that fascinated me, now, people, I usually talk with my guests beforehand, so I have an idea of their background and so on. I will tell you what is the standout piece of information for my audience to know. Sabine, you were the first to publish about this Z-Pack, which is ivermectin and hydroxychloroquine and zinc. Is that correct?
I was the first to find COVID in the stools. So we were the
Mm.
first lab to identify whole genome sequencing of COVID in the stools. Because when COVID hit, I was about to do a conference with a bunch of gastroenterologists and scientists in the microbiome space. and the head of the National Institute of Standards, Scott Jackson, and Howard Young from the NIH. So we had this whole meeting prepared in March, and then COVID hit, and we were all stranded at home. So me and Dr. Barodi basically put our heads together, and we started looking at all the protocols. And actually, we started way back in January because we were following the news and seeing what was going on in China. In March, I wrote a protocol that I submitted to the FDA, and it got approved April 2nd. It was the protocol on hydroxy, chloroquin, Z-Pak, vitamin C, D, and zinc as a treatment protocol. And then I had another one, which was the idea of let's say you're exposed to a patient with COVID and you don't want to catch COVID, maybe take 2 hydroxychloroquine and then vitamin C, D, and zinc just for that day that you're exposed, and then basically see what that does. So that got approved as a prophylaxis protocol. It was only 2 pills of hydroxychloroquine and then vitamin C, D, and zinc from there, right? So we got those 2 protocols approved early April. At the same time, I told, the NIH, because I was working with NIH and Scott Jackson at NIST, National Institute of Standard, which, by the way, if people don't know about NIST overlooks DARPA, they overlook CDC, they overlook the FDA. They're really the validation branch of the government that says, yes, this is accurate, this is not. So I work in close contact with Scott Jackson from NIST because I have a lab that needs to be validated as well when I write this. So I need to make sure that, I have their blessing in a way, right? So when COVID hit and I was discussing this with Scott Jackson and Howard Young and how to treat, right? Because I was right away on the front line. I told my scientist, I said, I bet you COVID is in the stools, because everything you put in your body, whether you put it in your nose, whether you put Botox in your, around your eyes, that ends up in your gut. We find that signature microbiome in the gut. So my first interest was, as I was writing the protocols to treat, to prophylax, I said, let's also look at stool samples. So I started collecting stool samples with my colleagues that were on the front line. Alan Miller, in particular, is a physician in Atlanta. And he started collecting stools. I started collecting stools. We had stools from Atlanta, we had stools from Beverly Hills and LA area and Ventura area. And then we started looking, and we found the whole entire genome sequence of COVID in the stools of patients that were positive for COVID. So that was
Let me just clarify something. So, in effect, when you put together this ivermectin, hydroxychloroquine, vitamin D, and zinc,
I put together hydroxychloroquine, Z-Pak, vitamin C, D, and zinc. And then I put together later on in August, ivermectin, doxycycline, zinc, vitamin C, D, because that was really Dr. Barodi's vision, was more of ivermectin. I was more following Didier Raoul, hydroxychloroquine, but I was looking at both of them. Somewhere along the line, I realized that I needed both drugs for the severe patients. That was never really a protocol that was submitted to the FDA. That was more me treating on the front line. Patient is crashing. He got put in one of my protocols. He desaturates. And I don't know if he got hydroxychloroquine or placebo. So I'm going to, give him ivermectin to try to treat him. So when I noticed that these patients that were on my hydroxychloroquine protocol were essentially crashing because probably they got placebo, because I was blinded. I didn't know who got placebo and who didn't. When they crashed, I had to think fast of what else to give them. So I was quick on the vitamins. I was quick on the ivermectin and then doxycycline. And then lo and behold, we noticed, wait a minute, these patients are doing great. And at some point, there were a couple patients that were really crashing that were on the ivermectin, doxycycline, zinc protocol. And again, I didn't know if they got ivermectin or doxycycline. So I switched and gave them hydroxy and Z-Pak. And then at the same time, I noticed like their oxygen was dropping. One of the key findings that I found during the pandemic was actually this concept of oxygen increasing when you give high dosages ivermectin. And that was really a key finding on the front line. I had a patient that had an oxygen in the 60s, 70s, went to the hospital, and I gave him 36 milligrams on a whim of ivermectin, not really thinking, okay, well, I was thinking he was going to the hospital and he was going to be admitted and probably die. So I said, why don't you stop by McDonald's, get some French fries, because fat absorbs the ivermectin better. And then all of a sudden he gets to the hospital and his oxygen is in the 90s. So the doctor said,
Wow.
go back to Dr. Hazan. She seems to know what she's doing, and just get treated. And this guy survived, by the way, severely hypoxic patient, morbid
So
disease, diabetic, etc. So it was a teaching lesson. So when I had these severe patients, I gave them hydroxy, Z-Pak to kill the virus, because we had an assay of COVID in the stool, so we knew that it killed the virus. But then we gave them ivermectin, vitamin C and D, and zinc to increase the microbiome. That was the concept.
yeah. So listen, you therefore were doing this even before Dr. Zelenko, and therefore you would have been well, they would have known about your research. Was this, or let me put it this way, there was a point where the president said something about HCQ, and the minute he said that, it became political.
Yeah, that was,
What were your thoughts?
the first thing I did is I looked up to the sky and I said, why me, God? That's what I said. That was my thought. Why me? Because I realized this was my protocol, right? That was attacked. Yes, Dr. Zelenko was simultaneously treating, because he looked up the data. And so did Dr. Peter McCullough, by the way, who basically also treated, looking at the protocol of Didier Raoult. You have to understand, at the beginning, we didn't have much. We had, what was France doing? What was Italy doing? What was China doing, right? So I was privileged, because I own a CRO, a clinical research organization, that I could join up with all the doctors around the world. So I could call China and say, what are you guys doing that's helping your patients? I could call Italy and say, what are you guys doing that's working, that's treating your patients? The vitamin D concept actually came from Italy. And same with the vitamin C. When I saw that some hospital patients were discharged on IV infusion of vitamin C, I'm like, that's got to play a role. And I'm not a person that just gives one thing. I'm a person, I was trained by Dr. Barodi and all my GI colleagues, that when you treat a bug, you don't just give one thing. You have to treat giving multiple combination. How did we treat H. pylori? Combination therapy. How did we treat hepatitis C? Combination therapy. How are we treating, prophylaxing, or treating HIV? Combination therapy. So the idea that, just vitamin C. Yeah, vitamin C may work on some people. Vitamin D may work on some people. Ivermectin by itself may work on some people. But guess what? If you want to treat the patient, you have to look at the patient. You have to take the history, and you have to do combinations. It's like a recipe. So, you're creating that recipe for that patient. And that's basically what I did during the pandemic, is use these key components and use them for the patient, right?
This is so important, it's I was saying just recently to somebody, it's like heart failure. There's no protocol for heart failure. You have to use a toolbox, and depending on the patient, and it's the same principles. When you understand what you're doing, you then can use the tools to make sure you get your specific patient better. That's proper clinical medicine. That's tremendous. So this is why I brought this up, in the sense that I need people to know that when you are talking now, you are talking from 4 years of research into what the heck was going on. And therefore, that led you into this question about the microbiome, because you guys first identified that the virus was in stool.
Yes.
What then, when did you start to notice that not only was the virus in stool, but it was affecting the microbiome?
So is the virus affecting the microbiome, or is the treatment affecting the microbiome, right? That's the question we're going to be answering. The other question is, did the virus penetrate because of loss of good microbiome, or did the virus caused the loss of good microbiome? That's another answer we're going to be publishing, hopefully in a world that's, less corrupt. And I say that because unfortunately I've discovered that the journals are captured, and it's very difficult to publish when you go against the national public health narrative. And here's the problem with that, because I know something that no one knows. I can't publish it. And I'm not going to publish that until, research is a story. It's a story of discovering microbes, discovering what kills the microbes, what enhances other microbes. You have to start at the beginning. I cannot tell you about hydroxychloroquine and Z-Pak eradicating the microbiome and the virus until I tell you about finding the virus in the stools. That paper of finding the virus in the stools took 6 months to publish at a time where everybody was dying. Is that appropriate for a paper of finding a virus in the stool, whole genome? This was an instrumental paper because this told the GI doctors, hey guys, you need to start focusing in the gut. Your patient may not get better because he probably has COVID in the stools. And here's the problem with that is if you have a case of a long hauler, right? Is it long hauler because they've demolished their microbiome? Or is it long hauler because they still have COVID in the stools, right?
Yeah, these are some important questions, Sabine. And you're quite right. I mean, when I was doing the publication on autoimmunity, it took us 9 months to get through. Now, this, in my view, was absolutely critical to understand. Now, in your case, you said something that's very important, which is that the journals are captured. What do you mean, and what does that mean to the public? Because they just can't understand, or even actually clinicians and researchers think to themselves, if it's not published, it meant it wasn't good. They cannot understand the political or financial weight that influences this. What is going on?
Financial. It's financial. The journals are getting paid by pharmaceutical companies to advertise. And unfortunately, that money that supports these journals biases them. There's a huge bias in healthcare based on the price of a stock of a drug. And I said that when I spoke at the Senate hearing with Senator Ron Johnson. The price of the stock is what we cared about during the pandemic. We didn't care about the price of a life. We cared about how high can we get the stock of Moderna and Pfizer go up, right? Before then it started crashing, right? So unfortunately, when you are biased by money as a journal, when you are biased by money as an academic center and you're going to lose your funding, you have to stick to kind of push the narrative of the one that pays you the bills. And this is what we're seeing in medicine. That should not happen. We should be allowed to publish both sides of the narrative. Research is about being controversial, right? Research is about going against the narrative, right? Because otherwise we're never going to learn. If we just follow, one person that has one vision, one hypothesis, we're all going to end up down the rabbit hole and we're going to kill humanity. That's what I saw during this pandemic. That's why I stepped in. That's why I did my research. You have to remember, nobody paid me to do my research. I didn't receive any grants from anybody. I did my research. I jumped into it myself. I started collecting stool samples of patients myself. I started analyzing these stool samples. These stool samples cost above $3,000 to analyze, to look for COVID, to look for the microbiome, to look at the reproduction of the microbes. This was not an easy research. Plus, supporting, employees that are working in the lab with with COVID that is possibly could kill them, could possibly go to their families and bring home the virus to their families. This was not easy. So for me to step into this really was because I saw that my voice was censored. I saw that my research on hydroxychloroquine was blocked, interfered with. I saw that my research on ivermectin, doxycycline was interfered with when all the celebrities started talking about it and there was calling it a horse paste. Who's going to join a clinical trial when they are being told you're going to be receiving a horse pace? Who's going to join a clinical trial when they believe that hydroxychloroquine is going to kill them, right? I had a patient with heart disease who refused to take hydroxychloroquine because he was biased by the media and therefore ended up in the hospital and died. He didn't want to take the drug that I gave him, right? So this was interference with research affects everyone. And here's the thing. Right now, I stand on very important information that I cannot share with the world. So yeah, there is a, when my hypothesis, I had a hypothesis of ivermectin increases the bifidobacteria. It was retracted after 8 months of being the most read paper in the journal. And by the way, I was the editor on the microbiome in that journal. Okay. And they retracted that hypothesis because it didn't fit with the narrative. It gave ideas to doctors that, hey, ivermectin could work by increasing bifidobacteria. And maybe that's why some doctors are seeing it working maybe in colon cancer. So this is basically what happened, right? So censorship of research, retraction of a hypothesis, woke me up to the corruption and the fact that the journals are captured.
Listen, I mean, that is a brilliant. I hear so many people have lost faith in science. They've lost faith in medicine. And I think in just that one series of statements you've just made, you may have saved medicine for the people who hear what you've just said, because that is pure science. That is pure medicine. You just want an answer. You just want to save lives. You're not interested in the politics. You're not fussing about the stock price. You're just trying to find an answer. Now, in that context, then, because where we are now, ivermectin, they're only starting to pull back on the criticism of it with what happened with the FDA, changing. So people are now starting to actually think a little bit more about it. How much have we lost by ignoring or not, I should say, not researching why it could help? This is the question. People just say, well, it can't work because it's an antiparasitic, instead of saying, well, actually, if it does help, what could be the mechanisms?
Right. And by the way, treatment works. Early treatment works. I've lost no one during the pandemic. No one died. So, and I treated some of the most hypoxic patients. In fact, I published a paper on multi-drug therapy in hypoxic patients treated at home. Okay. So treatment works. I treated, a high official politician in South America. I treated people in England. I treated people, high executives, celebrities, millionaires, billionaires, and no one died. No one. So, and I'm a Malibu physician. So believe me, if one of my patients crashed and died, I'd be on People magazine. So no one died. That tells you right away that interference with research happened and that people didn't really get to understand this virus very well. I think not to kind of, say, I'm doing research. I did, and you said it yourself. You said, you did research for the right reasons, not to be political, not for the price of a stock. Let me just tell you why I did research. I did research for me first. I needed to understand this virus before it attacks me. I did research for my kids, for my husband, for my parents. I wanted to understand what this virus was doing and what the vaccine was doing too, right? Before, so that I can make, as a scientist that's been doing 30 years of clinical trials for pharma, as a scientist, I wanted to do my own research for me first. Because ultimately, when you interfere with the research and you get a different answer because you're trying to make money, the only person you're going to affect is you, because you're going to be the patient because you interfered with research to try to make money. Money is not going to save you. Trust me, there's so many billionaires that have cancer right now and they're dying to have some kind of answer. They will give all their money to get another year or two to live with their kids. So money doesn't save you. Good research, unbiased research, non-political research will save you. And that's what I tried to do this pandemic. And
Yes, that's
I could be wrong. But guess what? Here's my statement to everybody that criticized me. Prove me wrong. Prove me wrong. That's it. You don't have to retract my paper. Prove me wrong. I proved that ivermectin increased the bifidobacteria, and I presented it at the American College of Gastro as an abstract. Okay? It's yet to be published. But guess what? Prove me wrong. Do the experiment.
Wow. So yeah, so this is important stuff, Sabine. I mean, we haven't even touched on the microbiome yet, and this is so fascinating to listen to. But before we even go to the microbiome, what were your thoughts with that kind of work with early treatment? What were your thoughts when you saw the transition of the world where suddenly all the eggs are put in one basket? Everything else was stopped, no treatment. Everybody just needs to be inoculated. What were your thoughts?
Well, my first thought is I don't trust anyone anymore. So that was my first thing, because remember, I'm in the clinical trial business. I brought vaccines to market. And when I saw the research being done poorly, that was one of the big red stop for me. So yeah, my thoughts were I don't trust anyone, and I definitely don't trust these vaccines because the research is done improperly. And why would they stop early treatment? Isn't our mission as physicians, as scientists, to save as many lives as possible? Why would you stop a treatment? My idea during the pandemic is put everything on the fire, do the vaccines for the high risk, do the treatment, put everything, like just turn off the fire, stop the virus in its motion, right? It didn't happen. Unfortunately, this virus did mutate. It got up to Delta, and then eventually Omicron came around, and that was the big savior, in my opinion, because it kind of like made us realize, wait a minute, we have a natural virus that's created out of an unnatural one in a way. So this was how the pandemic occurred, and it's a shame that they didn't just want to do all of it together. And then you go back, when you have a fire, you use all sorts of buckets with water, hoses. You don't say, oh, this one has a better high pressure. You just turn off the fire, and then you look back and you say, the high pressure hose turned off a lot of houses from burning than the bucket, right? That's what should have happened. It should have been a retrospective study in the sense that, hey, doctors, here's a protocol. Do your protocol, innovate. You're out there on the field treating patients. We trust you. You're a patient-doctor relationship. When we talk about hydroxychloroquine and Z-Pak, I wrote the protocol. We had 2 pages of medications that were contraindicated with hydroxychloroquine. So if you don't take the history properly to look at the medications that are contraindicated, you're giving it to the patient. You're not going to achieve success. You have to do due diligence. On the same side with ivermectin, we had another 2 pages, well, actually 1 page of drugs that were contraindicated, right? So you have to take that into consideration when you treat a patient. And therefore, medicine happens on the front line between a patient and a doctor. What should have happened is it should have been a lot of different protocols. Whoever wrote the protocols, put them out there. The government should have stepped in and said, look, here are all the protocols that we approved. And, feel free as doctors to evaluate and start treating and stop the virus from going on. But instead, they suppressed early treatment because there was a price of a stock that was behind it, okay, to sell the vaccines. And then when they couldn't sell the vaccine because of the fact that people lost trust and people got COVID, after they got vaccinated, and they saw others that weren't vaccinated still standing, still not having COVID, then their light bulb started, the lack of trust started happening, right? For me, the lack of trust happened from the beginning because I'm in the clinical trial field. When I see a patient is given a vaccine in a pharmacy and it's a kid, and we're not even enrolling in the kid's trial yet, and there's no informed consent given to that kid, and the kid gets a vaccine in a pharmacy, we have a problem. I had a problem with that because I said, we're vaccinating everyone. Nobody's getting proper informed consent. I had a friend of mine who's a photographer, had a coagulopathy after the vaccine. She was on birth control pill. Now, were we supposed to be giving birth control pill with the vaccine? Good question, right? But certainly, had this woman read informed consent, knew about the coagulopathies from the vaccines, right? Even though minimal, she would have probably made a different choice for her, because now she's on blood thinners for the rest of her life. So this is the problem. Informed consent was not given. Suppression of science, interference with research happened. And unfortunately, that affects everyone.
Yeah, this is some huge statements, Sabine, because the damage that has been done to the credibility of science and medicine is humongous. I mean, I keep on warning people. I'm saying to them, listen, some brown stuff is going to hit the fan. And when it does, if you are not standing up and advocating for patients, they will never trust you again.
And the
This
brown
is
stuff is hitting the fan because the microbiome is in capsules right now. So yes,
absolutely.
if we don't do the research, if we don't stand up right now to understand the research better, we are going to get stuck with a lot of problems down the road, especially
So let
with microbes.
All right, so let's talk a bit about the microbes now. In terms of people just don't think about the microbes. The microbes are, just brown stuff that falls in the toilet. Why is it important?
Because we've come to realize that if you don't have a solid microbiome, a balanced microbiome, you are susceptible to viruses. You are susceptible to Clostridium difficile, that bacteria that causes diarrhea and kills people. So the only treatment for that is really to implant microbes, right? I spent 30 years trying to kill C. diff with medications, antibiotics, and then clinical trials. When I couldn't fix my patients with C. diff with clinical trials, I would do this procedure called fecal transplant, the process of taking stools and putting it into the colon. What I discovered through fecal transplant was that these patients with C. diff had loss of diversity in their microbiome. Very few phylum were left, very few, a lot of good microbes were lost. And so the answer was really transplanting a lot of good microbes, right? Re-fertilizing your soil, right? So I'm a huge gardener, as I told you before. And one of the things that I equate the microbiome is really to that compost bin that's in our colon, right? So when you look at a compost, what are you doing when you're composting? You're putting green vegetables and bacteria together and you're mixing it and you get that brown dirt. That's the same thing that happens when you have that brown poop coming into your toilet. You've essentially given food to your gut and you have microbes in your gut that are metabolizing, that are decomposing those green vegetables, that food, and then it comes back as a brown, material, right? But it recirculates, it goes into the planet, it refeeds the soil, it goes back under the ground. So, we are just a reservoir for microbes. And, when you start seeing the power of the microbiome, when you start, fixing a patient, and this was really a individual patient that I fixed, had C. diff, had chronic UTI, chronic psoriasis, and was suicidal. When you use a donor, and I used, Dr. Karutz, who's the stool bank at University of Minnesota's donor, because he basically screens them properly. When I used that donor, my patient suicidal ideation resolved. His chronic UTI resolved. His C. diff resolved. His psoriasis improved. He is now 4 years out and he's amazing. Okay. So the right donor for this guy was instrumental at changing his suicidal ideation. So when you see that, you start saying, wait a minute, I'm doing something there. I'm changing the microbiome and I'm changing the personality, right? We just published and just submitted data at the anxiety meeting in Boston, where we presented an anxiety microbiome, right? Microbes that could possibly be the culprit of anxiety. Now, is it anxiety triggering these microbes to grow, or is it those microbes are grown triggering the anxiety? But definitely there is a personality microbiome relationship. There is a gut brain axis that is occurring. So yes, the microbiome is very important. And this was the paper that we presented, where we showed there's a signature microbiome in anxiety patients compared to healthy controls. How did we do this study? We did so if you'll recall, during the pandemic, a lot of patients with severe COVID were very, very anxious. So we discovered this fingerprint microbiome in these patients. From there, we decided, well, we're going to start giving GAD questionnaires, which is like an anxiety questionnaire, to all our patients. And we're going to see if that microbiome changes with anxiety. And sure enough, the people that had a high anxiety score had high signature microbiome, and the people that had low anxiety score had low, microbiome signature. So we're coming out with an assay, hopefully, for anxiety that's going to help the psychiatrists. And now I'm actually working with psychiatrists at Yale. I'm working with psychiatrists at USC, UCLA, to basically try to find answers, because now we have a marker that can tell us, hey, this is a picture of what an anxiety microbiome looks like. Let's try this. Let's try this drug. Let's try this drug. See if it improves the anxiety and improves the microbiome. That's ultimately how, the best research is done, right? You have two different set points that basically show you treatment. When I treat my patients with Crohn's disease, I do, an activity index to see how severe is their Crohn's. Then I look at their colonoscopy, and then I mark that, how severe is their Crohn's. Then I look at their microbiome and see how bad is the microbiome in these Crohn's patients. Then whatever I do as far as treatment, whether it be a biologic, whether it be, an antibiotic, multi-antibiotic treatment, fecal transplant, whatever treatment, bovine immunoglobulins, whatever treatment I do, I have to improve that activity index. I have to improve the colon. I have to improve the fecal calprotectin, which is a third marker. And then I have to improve the gut microbiome. This is how you do research. This is how you do science. Science that's well done is reproducing not only on a symptomatology way, but on a validated lab assay, and then on a clinical picture of the colon, and then on a microbiome picture.
Wow. Brilliant, Sabine. Brilliant. So listen, so taking you now, before we talk about the issue of what you found with vaccines and the microbiome, I'm starting with the work you had done, but this was a paper from China. They found that the gut microbiome changed with infection. So this is with infection. And they were looking at multiple different changes with specific bacteria that occurred, similar to what you found. What bacteria is primarily damaged, and is it a good one or a bad one?
So in the microbiome space, there's good or bad. It's really about the balance. So I like to, not all, it's kind of like humans. Humans are good and bad, right? So microbes are the same. You could have E. coli, that's a good microbe, and you could have E. coli, that's a bad microbe. You could have Clostridium difficile, that's a good microbe just staying around, being dormant and doing what it needs to do. Or you can have Clostridium difficile secreting its toxin, and therefore killing the host, right? So it's really about the balance. Now, the most important that I found, and really it was a path of discovery, was bifidobacteria. When we looked at COVID in the stools, We identified COVID, we identified different strains of the virus in different people, and therefore different mutations at different levels of the virus, which really in my mind was, how do we vaccinate someone when you have like 34 mutations in the spike protein, right? And how do you vaccinate someone? You're only vaccinating with one spike, right? So, and it's always the same kind of spike. So are you really vaccinating? And is that the way, should you even vaccinate against a mutating virus? Or should you even vaccinate against a virus, period? Right? So the idea of vaccination came from, bacteria. You mimic the bacteria. It's easier to mimic a bacteria because a bacteria, if you look at the bacteria, the genome, the gene of the bacteria is like ABCD, right? ABCD is ABCD. So if I take a vaccine that's ABCD, it's going to mimic that bacteria. The problem with the virus is that it's not necessarily always ABCD. It's ABCD 1 year. The next year, it's the next infection, it's ABDE. It's AFGE. It mutates, it changes. Viruses lack the capacity to clean up the mutations, right? Bacteria have properties that clean up the mutation, right? So you always have ABCD and you can always identify the bacteria. The virus, it's not so simple. So when you have AFGE and you're giving a vaccine that's ABCD, well, you've missed the genome. You know what I mean? So you've not stopped. So should we really vaccinate viruses that are mutating constantly? I don't believe so. I don't think so. I think viruses need to be suppressed by bacteria. Bacteria is bigger than a virus. So what we discovered with COVID is really that when people had severe COVID, they had a loss of a microbe called bifidobacteria. Bifidobacteria is this strong, potent bacteria that appears in newborns and disappears in old people. When I looked at my data early on in the pandemic, before COVID, I started looking at why are the old people affected, the diabetics, the obese patients, why are they affected? What's in their microbiome? The one significant difference was the loss of bifidobacteria. Why are the newborns not affected? The one significant difference was the high bifidobacteria. So with that idea, knowing that data that I didn't publish, right, that I had in my data bank, in my clinical data bank, in my lab, I started saying, okay, well now let me look at the bifidobacteria in families. Let me look at the families, the kid that has COVID, severe COVID, and the parents that didn't catch COVID. Why did that happen? What is different in the microbiome of the family where one kid has COVID and the rest doesn't? And lo and behold, we discovered bifidobacteria. So we actually published pre-existing signature microbiome in COVID because we discovered that bifidobacteria. So when I discovered that in that one family, I said, let me look at another family. So we started looking at another family and we discovered the baby had no COVID, had a lot of bifidobacteria. The 5 family members had 0 bifidobacteria. We took that on a bigger level and said 72 patients. Now remember, every time I analyze these stool samples, it's $3,000, right? So, for me to do a 72 patient study where I'm collecting stools from highly exposed patients compared to severe patients, and I'm showing the bifidobacteria, that's an expensive study. So for that paper to take 8 months to get published, I had a problem with that because that was a hint during the pandemic of how to survive COVID.
Wow. No, listen. So, in effect, then, are you suggesting that well, I mean, the microbiome did change pre and post infection, even in people who had bifidobacteria, say, what would be the mechanism for that change? Why would that genus be affected?
did the virus create a bacteriophage? Did the spike protein create a bacteriophage? That's the biggest question, right? So we're going to be lighting some, when I'm able to publish, we're going to showcase how the virus comes in to the microbiome, what microbes are altered, and whether treatment affects the microbiome or whether the virus, the spike protein itself, affects the microbiome. So
Now,
that's a
just in,
study that needs to be published.
and now, just to clarify for the audience, when you said bacteriophage, these are viruses that infect bacteria,
Correct.
similar to like what would happen with diphtheria. A bacteriophage causes some of them to produce a toxin, which can
Correct.
then cause disease. Now, here is the question, Sabine.
I like to think of it as a balloon full of toxic material, and you have a virus that has a spike and basically pops the balloon and the toxins come out. That's how you
Okay,
have bacteria.
so here is the other question then. Is it possible that the virus alters the microbiome so that certain bacteria get capabilities that kill other bacteria? Is that a possibility?
It's a possibility, yes. It's a possibility. And it's a
Now,
possibility that the virus brings on other viruses as well. We tend to
wow,
think of just bacteria, but think about it. It's a war. It's a war between your gut microbiome, your gut bacteria, which you're supposed to have a lot of, and viruses that are like 1/20 of the size of a bacteria. So these viruses are penetrating. You've let the door. You've opened. When we talk about leaky gut, what are we talking about when we talk about leaky gut? It's really an imbalance between the good bacteria and the bad bacteria. That wall is open. Viruses are going to penetrate. Bacteria with toxins are going to penetrate. They're going to go through the wall. They're going to travel the nerve. They're going to go in the blood vessels. They're going to go in your bladder. They're going to go in your uterus. They travel. They're going to go to your heart. The body, we tend to think, which was the idiot thing of the pandemic, which really made me lose faith in medicine, not medicine, but science, was the narrative and the allowing a narrative that says the vaccine stays in your deltoid. No, the vaccine does not stay in your deltoid. The vaccine goes into your blood vessel, your muscle, your nerve, your lymph nodes, your lymphatics. It circulates. Everything circulates. The idea that something stays in one place. What I told you, when I analyzed the microbiome of women that got Botox in their eyes, around their eyes, I found botulinum toxin in their guts. Everything circulates.
absolutely. So here is a controversial question that I think needs to be addressed from a scientific point of view. So forget the politics, but we still cannot identify the origin of the virus in terms of which natural vector. If by chance this was made in a lab with regards to these characteristics, where a virus that infects a human is also able to penetrate bacteria, is extremely unusual. Is that conspiracy stuff or what?
It's not well, you could make it conspiracy, and that's what they're using to kind of discredit those that are coming out with the ideas. And you could say, oh, for what purpose? To kill humanity? Conspiracy theory. I don't go that rabbit hole. My idea is basically, listen, scientists come up with ideas every day. The other day I read of a scientist that is planning to reproduce a dinosaur. Why in heaven's name would you want to reproduce a dinosaur? To what? To kill humanity? I mean, that is the stupidest experiment in the history of experiments. So unfortunately, you've got these scientists that come up with these ideas. They become friends, with others. They're probably smoking something and they're like, hey, let's raise some money and create a dinosaur, right? And then money comes in and then the stock market goes up to create a dinosaur. But they don't think about, well, what the hell is the dinosaur going to do to my backyard, to my house? Is it going to kill my kids? How am I going to fight a dinosaur? And is that dinosaur going to reproduce? And what's going to happen to the microbiome of that dinosaur on the planet? Is that going to kill the microbiome of the planet? So just because you can do an experiment doesn't mean you should do it. Okay. So the problem with this lab or whatever happened is some scientists decided probably, and I'm going to say probably because I've seen the genetics of the virus entirely. Some scientists decided, hey, and we have the capability right now, by the way, to cut these bacteria, to cut these viruses and to join them together. So is that what happened? Was a virus cut and a bacteria that can enter a cell without a cell membrane? Can the joining of a virus and a bacteria create enough havoc in the gut? And therefore, is the concept of the vaccine a piece of a bacteria to try to offset, but it went wrong because unfortunately, that bacteria that penetrated the cell wall utilizes probably the genetic material of the cell wall and reproduced itself. So again, scientists with poor ideas that were not really thought of. Every time you CRISPR something, every time you join one thing with another, you're creating an unnatural microbe. What do you think the microbiome will do when it sees an unnatural microbe? It will reject it, because our bodies are designed to reject the unnatural. Think, or the overly bacteria, right? If I take a hamburger with a lot of E. coli, what's my body going to do? It's going to have diarrhea because it's rejecting, right? When you have an autoimmune, and you published on autoimmunity, when you have an autoimmune response, that's your body rejecting the bad stuff or the unnatural stuff. So we need to stay, human beings are trying, and I get it, right? Trying to CRISPR the gene, trying to CRISPR the microbes, but we've yet to reproduce blood. How about we start with reproducing blood so we don't need blood transfusions from human beings constantly? We constantly need human beings to give us blood. So now we're entering a world where we have altered the microbiome. There is not enough good donors in the world to fix that problem. So if we ever get to a world where we are, think about blood transfusion. And I always equate the microbiome to hemoglobin, right? When we had patients bleeding back in the days, we didn't have a hemoglobin to tell us how much should we transfuse a person, right? Hemoglobin is 7. I need to transfuse the person because they're below 7. Now we have all that. And now we also know through research that was not interfered with, we also know that if you transfuse someone with hepatitis C, you're going to give hepatitis C to the patient. If you transfuse someone with HIV, you're going to give HIV to the patient, right? So we've learned how to clean the blood as best we can, right? There's still a lot of stuff we don't know in the blood that we are transmitting, right? Viruses, for example. So when we are giving a blood transfusion, are we transmitting viruses that we don't know, therefore causing the problems in the patients down the road, maybe neurological problems, etc.? So these are the questions that need to be answered. So now with the microbiome, there will come a time where we will have the equivalent of the hemoglobin, and we will say, you, sir, have an autoimmune process, you, sir, have cancer, and you are lacking these microbes, and these are the microbes you need, and here's a transplant, right? And maybe it may not be a transplant, it may be a consortium of microbes, okay? But here's the thing. What if you kill all of the microbiome of humanity? Where are you gonna get your good donors, right? If you transfuse everyone with the same blood, where are you going to get your good blood transfusions, right? So
Yeah, so it leads right into this question then, because not only are we seeing changes in the microbiome with infection, but your paper, the one that they don't want to publish, it's that you're seeing it as well around the vaccination. How can you be sure that it's related to the vaccination and not something else?
You can't be sure of anything in science. It's about, like I said, it's about prove me wrong. Okay. Add something else to my data. Okay. And this is still continuous data, right? We're still looking at the vaccines. When we got censored from doing research on ivermectin, doxycycline, zinc, and we couldn't push our protocols, to the next level, or the hydroxychloroquine, and the push of the vaccines. And really, for me, I wanted to know, because the whole time I was like, what is that doing to my microbiome? Because you have to understand, Philip, before the pandemic, I was conducting 50, and I'm still conducting 57 clinical trials on the microbiome and diseases. I'm looking at the microbiome in Parkinson's. I'm looking at the microbiome in Alzheimer's. I'm looking at the microbiome of GIST tumors. We just picked up some, microbes that could be linked with GIST tumors. The microbiome of colon cancer, appendiceal cancer. So I have a vision, a small vision, but still a little vision of what is going on in the microbiome, right? With my path, right? And especially following the bifidobacteria. We noticed bifidobacteria was absent in Lyme disease, Crohn's disease, invasive cancer. So all that becomes important when you kind of say, okay, well, we know these diseases share these things in common. How do we fix this, right? So when we looked at the vaccines before and after, believe me, I did not want to be the person that came out and said, oh, vaccines affect the bifidobacteria. I knew I was right about it for two reasons, okay? Those that came to me that were vaccine injured, people that I trusted, my colleague, gastroenterologist at Cedars-Sinai in Santa Monica, top of her game, treated like half the celebrities in Beverly Hills. This woman gets vaccinated, all of a sudden is debilitated, okay? She went to the top neurologist at UCLA. He called me and said, can you look at her microbiome? When I saw she had 0 bifidobacteria, 0 actinobacteria, I was like, okay, well, did the vaccine do this? Did the spike protein create a bacteriophage and kill the bifidobacteria? Let me see more. Then I started looking at the kid that was paraplegic and her brother. Again, the kid had 0 actinobacteria phylum, 0 bifidobacteria. Her brother had some bifidobacteria. He got the placebo. Then I started looking at more vaccine injured. None of this can be published, by the way. Nobody wants to hear about it. We have hundreds of patients that are vaccine injured that volunteered for my study, that actually, sponsored or helped us with funds to see their microbiome, okay? And all of them have 0 bifidobacteria. So when you look at those patients that have 0 bifidobacteria, and you look at my data where it was before and after, you have one guy that was like at 1 million bif and drops to 0, and then persists in dropping, okay? You have to ask yourself, is the spike protein creating a bacteriophage? Is that spike protein, and remember, it's not just the spike protein, it's the spike protein with the nanoparticle. It's the spike protein with the contamination. It's the plasmid. It's all that. Is that going into the blood, into the circulation, into the lymphatics, into the nerves, into the colon, and landing there, and now utilizing the properties or other microbes in the microbiome space in the gut and reproducing? And now you have a leaky gut, and now all these microbes are passing, right? Is that being created in the gut, right? Are you having a spike enter the blood, going to the colon, utilizing properties of other microbes, reinforcing that spike, right? Or even that spike triggering another virus, triggering another bacteria.
So,
We don't know.
this really leads into an important paper at Carlo Brogna in Italy. What he found is that after infection with the virus in the stool, they were finding up to 36 different new bacterial toxins being produced. So, a question to link the two together: could it be that the spike protein mRNA, so the mRNA, not necessarily the spike protein, is picked up by certain bacteria these bacteria can read it maybe in a different way than the human body, produce toxins that kill the other bacteria. Is that possible?
That's possible. Absolutely. I mean, we're in a world that, we've not let a loose spike protein, right? The one thing that the coronavirus has that's different from other coronaviruses is that spike protein, right? Whether that was an experiment that inserted it or whether it got inserted on its own, I don't know. I'm not going to go down that rabbit path. But the fact is, the spike protein is the problem. Spike protein injury is what's causing so here with the long haulers, the virus, is it the spike protein that is causing the problem and still going in the gut and in the blood, etc., or is it that the virus is still lingering in there, or is it that they have a desolate gut, no microbes to fight anything, and therefore everything is toxic, right?
Wow. So when we look at where we are now, okay, so let's see if we can tie this together, because this is some heavy stuff that we're taking on. Firstly, how are you going to get the scientific community to actually even just look at this? Because at the moment, as you said, it's like there is only a narrative. Nobody wants to come outside the box. No matter what is happening, people are locked in the box. They don't want to come out. How are you even going to get this kind of information out there?
The same way that I got hydroxychloroquin and ivermectin to go out there, word of mouth PR. I've been in the clinical trial business for 30 years. I know how to get the word out there. I was the number one recruiter for C. diff for clinical trials. I know how to get the word out there. The one thing we have as physicians is we kind of collaborate, and that collaboration is the most beautiful thing. What we saw during the pandemic is about 300 doctors collaborating together to treat humanity, and we all joined up. We all, even though we had different ideas, Pierre Corey was on ivermectin, Peter McCullough was on hydroxychloroquine, but eventually they saw that it was not about their protocol. Didier Raoult was on hydroxychloroquine. They all saw it was a bigger force trying to get us out of the picture almost, right? They're trying to remove the idea of physicians and make us like the bad guys. Oh, those doctors didn't save us during COVID. Our hands were tied. Think about what I had to do to save patients. I had to write a protocol, which cost me over $300,000 to put through the FDA, right? This is not cheap to write 3 protocols to submit to the FDA and to run these protocols so that when the Department of Health came to my office and said, well, Dr. Hazan, why did you treat COVID? I have a protocol that's approved by the FDA. Nobody had that opportunity, right? When I did that to kind of like showcase to the doctors, hey guys, this is a protocol that I did, and look at what happened to me. I am, in a way, the person that doctors are looking at, because I'm demonstrating what is happening to our field, right? When a physician that has brought vaccines to market is not allowed to publish the data on vaccines affecting the bifidobacteria, we have a problem, right? So it's all about the discussions have been shut. What happened during the pandemic is doctors joined forces and started treating patients. When what's going to happen is, and what you're hearing now is the microbiome, because here's what happens. The word, it's the butterfly effect, right? Action leads to a reaction. I talk about bifidobacteria. There's a million companies out there selling probiotics. They want this data. They want to hear about it because they believe there's a million people out there that are naturopaths, that are herbalists, that are into natural foods, real organic milk, real organic fruits and vegetables. Those people listen to what I have to say. And then they tell, it doesn't have to be Sabine Hazan telling. They tell their friends and they tell their friends. It's a boomerang effect. When you said at the beginning, Dr. Zelenko had a protocol that he read from Dr. Raoult. He went on his own path as a lone cowboy into the public. And people say, well, aren't you upset that he talked about your? No, it's my protocol is the protocol of the world. It was given to the world. If I wanted to hide it, I would have called it the Haze D pack, and nobody would have known that it's hydroxychloroquine, Z-Pack, vitamin C, D, and zinc. If I wanted to hide ivermectin, doxycycline, I would have gone on clinicaltrials.gov and put Zyverdox, and I would have never mentioned ivermectin. Think about what is Paxlovid. Do you know what's in Paxlovid? Right?
No, well, yeah, this is interesting now. Yeah,
Well,
what?
okay.
Yeah.
Paxlovid is a secret formula. What is in Paxlovid? Do your homework. I'm not going to say it. But the bottom line is every product that goes into the market has a secret formula. I did not need to tell the secret of my formula when I talked about Zyverdox. I could have just called it Zyverdox. So when everybody on April 2nd, every doctor looked at clinicaltrials.gov to see what are the solutions, because they wanted to be on top of it for their patients. They saw my clinical trial was number 3 and number 4. Okay. When they saw my clinical trial of hydroxychloroquine, it went viral. Doctors started writing it. Doctors started keeping it in their closet. When in July of 2020, I put out Zyverdox, ivermectin, doxycycline. Again, doctors saw that protocol and it went viral. Ivermectin had a point. When Dr. Brody started talking about it in Australia, about ivermectin, and then went as all of a sudden he was a bad person because he's trying to raise money for a protocol to save people from COVID. Isn't that what pharmaceutical companies do? Don't they do PR to raise money for a protocol? But all of a sudden, because it came from Dr. Brody, he's the bad guy. And, oh, I'm sorry, he's just a grifter. He's just there to take advantage. But pharmaceutical companies are not grifters. They're not even doctors. They're not even on the front line. You know what killed me this pandemic was the fact that people that never touched a patient with COVID were talking about COVID as if they knew how to treat it. And the whole time I was like, step into my lab. Step into my lab. Come analyze the thousands of stool samples with COVID. Open that freezer full of COVID strains. Where's your guts now? You're talking like you're a big shot on television. Where's your guts? Come and step into my lab. Go touch a patient with COVID, you wimp, because that's what it was. That's how I felt. I felt like people that were talking and never touched the patients were wimps. Go touch a patient with COVID. Go expose yourself with COVID. Bring it to your family. Then talk to me about treatment doesn't work. And I'm
Wow.
very heated about this because that was my pet peeve the whole pandemic. And, it brought me back to HIV days where I was a resident in Miami Jackson Memorial Hospital treating HIV patients as best I could. And patients were vomiting blood all over me, putting their bloods all over the walls. And then you had these hoity-toity celebrities saying, oh, this for HIV. And the whole time I was like, why don't you come to the hospital and try to treat these folks? It's not that easy. So when they discredit the doctors and they call the doctors like, oh, these horrible doctors. Well, I'm the last of the Mohican because I spent my money to do the research. No doctor's going to do that. Okay. Everybody's waiting for their grants. Okay. I was ready to trash money to prove a point. And I'm that girl. I'm the girl that's going to throw $1 million just to show a point because that's who I am.
Wow.
And this pandemic is corruption of research.
Wow. Tremendous, Sabine. So listen, as we come towards the end now,
yes,
people are worried and they're thinking, oh my goodness, is my gut destroyed? Do I just start taking probiotics? What do I do? Because this is complex stuff. And this, you are opening a Pandora's box of, oh my God, how do we fix this?
You fix it by supporting the Microbiome Research Foundation.org. That's one, by helping us, because it is not easy. Once you've broken a glass, and I'm not going to be, giving you a protocol, the Hazan protocol. I'm not. You want to join our research, support us, support our research, please do. We are looking into it. We are looking into fixing it. It is not easy. Like I said, it's fixing a glass and you're trying to put this piece of glass together and you don't know what it looked like before. So, the more data we have, the more we can publish, and then also help us fight to allow us to publish. It's become this, it's a huge job. I hired people to help me with bringing funds to donate, etc. And everybody that comes to me is like, this is a forklift. I mean, this is enormous. We're fighting on so many levels. We're fighting the corruption of not publishing. So we have to hire legal to help us to go after the journals that have censored us. Okay, that's expensive. We have to conduct the research on a lot of people that, when you talk about these people that have had COVID, that have had long haulers, they ran out of money. They don't have the money to spend on expensive stool tests. We need their stools to understand the data. We need them to join us so we can treat them. When you talk about, vaccine injured, we need to pay attention to all that. So, to me, and here's the thing, and I'm going to give a light, I think we tend to be, looking at the darkness. I'm not going to let us look at the darkness. I'm going to look at the positive. And I'm going to look at the same way that I stepped into COVID, because I realized that only a small percentage of people died. The majority stayed alive. Only a small percentage of people have vaccine injuries. The majority are fine because they have a resilient microbiome. So don't panic, don't stress, don't go taking antibiotics, probiotics, doing all sorts of stuff. When we panic is when we aggravate our guts even more, because we do things that just keep aggravating. So you start off with a good microbiome that survived COVID and has had no problems with the vaccine. Keep at it. Stay doing what you're doing, because listen, it's not right or left, it's middle. And we have to come to middle. We have to come to acknowledge that there are some people that are hurt by the vaccine and vaccine injured. And we have to acknowledge that there are some people that are hurt by long haulers, by the spike protein. And we have to understand it. So in order to understand it, both sides need to come to the middle point. And it's not a competition, it's middle point. That's it.
Wow. Sabine, as I said, this has been wonderful. I've just sat and listened to you, and I hope the audience realizes that we are dealing here with a true researcher, a true clinician who is fighting and advocating for patients and across the world. I mean, all I can say is I hope everyone follows her line. Please find a way to support her, because as she said, lots of people cannot afford to be treated, and in the US, believe me, it's going to cost a fortune to do anything. So you do need to be supported. I wish you all the best
Thank
at
you.
your company. I think that that's a tremendous bit of work, and I think for the world, please keep fighting for all of us.
Thank you. Thank you. And I think it's also, I'm going to just leave with one thing. Let doctors be doctors. We train to be doctors. We need to be allowed to innovate. If we stop innovation in medicine, we stop medicine. And Lord help us when AI becomes our doctor, because AI is only fed what it needs to be fed. So it's going to look at data that is not necessarily accurate. And AI is not on the front line treating and touching patients. We need that patient doctor relationship again. We need to trust our doctors. We need to give our doctors a break and hold their hands, because these are difficult times for doctors, risking their lives on the front line and fighting all the political, agendas that basically stop them from giving treatment. So let doctors be doctors and save the biff. That's my platform.
Beautiful, beautiful. Thank you. Sabine, stay with me. I'll just let the outro. Thank you, everyone, for joining us. And look in the description for links and support Dr. Sabine Hazan. Have a great evening.